Good afternoon, everyone, or morning if you're on the West Coast. Thank you for joining us today. We are excited to bring you the first webinar in the NIA Data Linkage Program's 2025 webinar series. Today's webinar will feature the Long Life Family Study. Next slide. The agenda for today's call will start with a few brief introductions and webinar logistics, followed by a quick overview of the NIA Data Linkage Program, which then leads into our featured topic, the Long Life Family Study offering. At the conclusion of the presentation, the panel will hold a question and answer session. Next slide. By way of introduction, I am Susan Seifert, the Program Director for the NIA Data Linkage Program, and I am joined today by Mary Wazinski from the Long Life Family Study, as well as members of her team. Given the webinar format, our team is requesting that questions be submitted via the Q&A chat box, which can be found in the lower navigation pane. The panel will do their best to answer all the submitted questions, time permitting. Should there be any remaining questions, our team would be happy to follow up with you via email following today's session. Next slide. Now for a brief overview of the NIA Data Linkage Program. NIA established the linkage program to address three major challenges that researchers face relative to gaining access to CMS data for linkage to their study. The first challenge is the time and cost for researchers to access CMS data. To overcome this, NIA collaborated with CMS to structure this program in a way that is able to provide quicker access to CMS data without the researcher incurring any data fees. One condition, however, is that researchers must use the CMS linked data in a federally authorized system that complies with the federal data security requirements. Finally, NIA recognizes that not all researchers are experts in the use of CMS claims data. The linkage program offers knowledgeable CMS data support staff to assist researchers. Next. Building on these challenges, the primary goal is to facilitate high-impact research in the area of health and aging while minimizing the cost of data. Next slide. Our comprehensive CMS data inventory includes Medicare enrollment data, Parts A, B, C, and D data, the CMS assessment data, which features the MDS, OASIS, and IRF-PI, and we also offer access to Medicaid data. Next slide. The CMS and study data is delivered to the researcher in the linkage enclave, which is a set of secure workspaces where researchers are able to analyze CMS-linked study data. Each project gets a dedicated project environment, which is accessible to only authorized members of the research team. Next slide. The linkage enclave offers a variety of statistical software packages, such as MATLAB, Python, R, SAS, SPSS, and Stata. The enclave also features a proprietary tool called the Job Board, which is a load-balanced, high-speed server farm which can be utilized to run your statistical programs, often in substantially less time than running the program using local resources. Next slide. The enclave features a file transfer tool that allows transfer of files into and out of the enclave. Importing files is generally restricted to code and supplementary files that have been approved by the study team to use in conjunction with their data. Export requirements are aimed at supporting dissemination of your research findings and are a bit more strict in that the data export should be aggregate level data with no PII or PHI or small cells. Data transfers out of the enclave require review by both the NIA Data Linkage Program and the study that provides access to their data. Next slide. The linkage program offers a three-pronged support structure. The linkage contractor offers access to the CMS data as well as enclave support. NIA supports the researchers not only through its support of this program, but also through the NIA DUA review and approval process for CMS data. Each study team offers their study data to researchers and a wealth of information about using that data. Next slide. The linkage team can be accessed through email or via phone, Monday through Friday, 8 a.m. to 8 p.m. Eastern. I know that this is a very quick overview of the NIA Data Linkage Program, and should you have any questions about the program, my team is happy to take questions at the end of the presentation or follow up with you via email. Now, without further delay, I would like to introduce the Long Life Family Study team to continue today's presentation.
Thank you, Susan. Next slide, please. So my name is Mary Wachinski, and I am here representing the Long Life Family Study. This study has 4 field sites in the U.S. 3 of them are in the U.S. and 1 is in southern Denmark. We have Columbia, Boston, and Pittsburgh are the field sites. Southern Denmark University is the Danish field site, which is not included in CMS data. We also have the University of Minnesota helps us. They do all the blood assays, and Duke University is an analysis center. Myself and the team here, which is myself, Dr. Michael Provence, and Rosa Jin, are at the coordinating center at Washington University School of Medicine. Next slide, please. So I just wanted to give everybody a brief overview of what the Long Life Family Study is. It's a longevity and healthy aging family study. And when we say family study, it is a family study. These are groups of people that are from 2-generational families. It's been funded by the National Institute of Aging continuously since 2004. It is international collaborative, and our main focus is on the genetics and familial components of exceptional longevity and healthy aging. Despite that being our main goal, it is very phenotypically rich, and this is because longevity and healthy aging is one of the most complex phenotypes you will ever analyze. So we have lots and lots of data to analyze. As I said, our families are 2 and 3 generations, and we have genomic data as well as multi-omic data available in addition to the phenotype data. I've already gone through the sites and the blood labs, so we can go to the next slide, please. Our data is also longitudinal. We have up to 3 visits now on participants, and these are in-person visits, which are usually conducted in their homes. So all the information we have on these participants are from protocols that are standardized, and we have centrally trained people going into the participant's home. Because of that, they also need to have protocols that can be done in the home and the equipment that can be taken into the home. Our protocols cover various domains of healthy aging. We have a lot of physical performance and a large cognitive battery. We have obviously information on longevity and survival, cardiovascular disease, diabetes, anthropometry, activities of daily living, most of the types of variables you'll see in epidemiological studies. Additionally, we also have a lot of biomarkers from blood that we've assayed. So we have serum lipids, glucose, glycated hemoglobin, a complete blood count, and most of these are done at every visit. Some are visit dependent. We also have markers of inflammation like C-reactive protein and interleukin-6, telomere length, looking at longevity. Further, we've used their blood to do extensive omics. Most of these are at baseline, but some of the participants we have done longitudinal, and we're still working on that. So we have GWAS chip data at baseline. We perform linkage analysis, so we have the identity by descent matrices to help us do that. We have short read whole genome sequencing on everybody. Metabolomics is done on everybody. And then on a subset, we have transcriptomic data and methylomic data. Next slide, please. So as a background to our longitudinal data, I thought a timeline of how we have collected this data would be interesting. So as I said, we've had 3 longitudinal in-person visits. Visit 1 was from 2006 to 2009 to establish the cohort. We consider our probands the top generation and the offspring as the second generation. This was the baseline visit with extensive health measures and tests, and then we had recruited 539 families, which led to 4,953 participants. Visit 2 was approximately 8 years later, between 2014 and 2017. We added, during this visit, we kind of checked in with participants on all of the extensive health measures from visit 1, but we also added an assessment of artery buildup using a portable carotid ultrasound machine. And we added additional cognitive tests and introduced a digital pen to also collect more cognitive data over the testing. In visit 2, we had 2,904 participants that remained, and this was 478 probands and 2,426 offspring. And I can tell you in the next slide one of the reasons for that, but let me finish the timeline. So for visit 3 then, we just finished in June of 2024. This one began in 2020, which was a little bit unfortunate because it began during the COVID pandemic, which led to a lot of delays in the study. But here we also did everything we've done in visits 1 and 2, and we added a food frequency questionnaire to obtain dietary information. And we attempted to recruit some of the 3rd generation, so the grandchildren of the proband or the top generation. And this is where we started doing our multi-omic assays. Visit 3 has 2,200 participants. Further, every year after a participant is enrolled in the study, we try to contact them either via phone, and now we also have a online survey that they can do for an annual follow-up. So we do collect some information every year on the participants, and this is also how we find out, if the participant is alive or dead most of the time. Next slide, please. So as I said, visit 1 was in 2006 to 2009. These are highly selected families that are showing exceptional longevity. So as you can see, overall, if you look in the overall column where there's 539 families on the far right, we had 1,727 probands between 2006 and 2009, but their average age was over 90. In the offspring, we had 3,200, and their average age was around 60. The average family size was approximately 9, and this FLOSS score is how exceptional they are in terms of living to a healthy age in terms of the family. And we set a cutoff. They had to at least have a FLOSS score of 7, each family, to be in the study. And this means that roughly 1% of families in the Framingham Heart Study would be eligible, and 99% of them would not be eligible to be in this study. And that's how highly selected we really were. So in the 8 years between visit 1 and visit 2, if you look further down, as I said, we only had 478 probands. A lot of that is because 8 years later, these people would have been over 98 to even 100. So we had a lot of attrition and deaths attributed in the proband generation, which is why our numbers keep going down. They're just getting old, and we can't do much about that. Next slide, please. I also wanted to share, we have published our objectives and design paper in the Journals of Gerontology. And this will give very in-depth information about our study and heritability estimates of our cross-sectional and longitudinal phenotypes. Next slide, please. So in terms of what we've done so far, I've selected 3 broad research areas and papers that we have done. We do a lot of aging research. We are a study of aging and longevity. So our colleagues from Denmark have looked at mechanisms that underlie family aggregation of exceptional health and survival, and that's looking at 3 generations of the Danish data. We've also seen that there's a slower decline in cognitive processing speed, which is associated with our families that we don't see in the general population. We've also looked at the association between frailty and perceived physical and mental fatigability. And we've looked at protective effects of family longevity, especially over ages 100. Next slide, please. Another area of research we've been into is mortality research. And this one is a little bit more recent because we do have we are working on updating our mortality or adjudicating our mortality events through our M&M committee, and one site was a pilot for this data. So we have looked at how perceived physical fatigability is actually a predictor of all-cause mortality in our participants. And we've also looked at all-cause and cause-specific death in our sibling generation. We've also used the composite measure of physiologic dysregulation to predict mortality in our participants. Next slide, please. Finally, as I said, we are quite the genetic epidemiology study. So we have done a lot of research in genetics. Most recently, we found a new gene, MTUS2, which is associated with late onset Alzheimer's disease in our group. We've shown that the ApoE E2E2 variant is associated with a slower rate of cognitive decline with aging. And we have found a novel locus for gait speed decline with aging. And again, this has been our main focus over the years, these 3 broad areas. We have many more publications, but I just wanted to give people an idea of what we have used the data for thus far. Next slide, please. In terms of the data, visit 1 was previously shared via dbGaP, all the phenotypic data. And since visit 2 or visit 3 in 2020, we have been moving all data to the ELITE portal, which is run by Sage Bionetworks. And this includes all the phenotype data, all the omic data, all the pedigree data, everything except the CMS data. Next slide, please. So this is the study page for the Long Life Family Study on the ELITE portal. It has this study description, methods, and then it has a study data tab. Next slide, please. Under the study data tab, it says how you can access the data within the ELITE portal in terms of data use certificates, what you need to have, and what sort of IRB approval you need to obtain our data from the ELITE portal. Next slide, please. So in terms of the Long Life Family Study in the Linkage Enclave, as I said before, it's only US participants. So at visit 1, which is what we used for the enrollment, we had a total of 4,953 participants, but 1,263 were Danish. So we excluded those and leaving a US sample size of 3,690. However, our Finder file has a few less than that, and this is due to various consent preferences. Some of our participants did not consent for us to share their data and search in the Medicare data. We also had some withdrawals since visit 1 where they no longer want us to follow them. And we have some US participants that did not have a US address. They actually were affiliated with a US site, but they live internationally now, and they also do not have a social security number or Medicare beneficiary ID that we could link to them. I also want to note that we submitted our Finder file most recently in October, and we just are getting access to the data right now. So we haven't even processed this data yet, really, or looked at it. Our goal, though, is to use a lot of the CMS data to validate our morbidity events. So We have self-report of all these various diseases and conditions that participants report having, but it is self-report, and we want to be able to validate that and give a more accurate date of diagnosis for those, because everything is recall bias from the participants right now. Next slide, please. So of this 3631, we actually had really good match rates in the CMS data. Overall, we have a 98.7 match rate, which is wonderful. But if you look at those 65 and older, which is the majority of our data, it goes over slightly over 99%. So I feel like we have really strong presence of those participants in the CMS data. Next slide, please. Lastly, we are wanted to give you some information about data access, and this is kind of a work in progress, mostly because we are so new to the enclave and we are still trying to figure out the process. So right now for the phenotype data or the omics data, you would contact the Elite portal. For the enclave, it's probably going to be a combination of the ELFs or LLFS here at the DMCC and the Elite portal to establish a DUA with us. Then you're going to need to complete the DUA request package and submit it to the linkage enclave to establish your DUA with NIA. Once you receive their approval, you would have access to any data on LLFS for your research question in the enclave.